Monocytes are fairly distinctive. They have generally what's considered abundant cytoplasm, generally described as a gray lavender cytoplasm. The nucleus usually stands out against the cytoplasm as being particularly basophilic. The nucleus in monocytes is very characteristically indented on one side, so it's kidney bean shaped. And that shows it very well in this particular picture. But you have to remember that these are three-dimensional objects, and so that nucleus can rotate in the cell. So you won't always see that indentation. Usually you will, but not always. But you should you should always see a fairly abundant amount of cytoplasm. And the cells are fairly large. You know, they're 12, 14 micrometers diameter. They are phagocytic in nature, and so as their phagocytic machinery becomes turned on, their cytoplasm tends to become more vacuolated, and the cells will actually become larger as that happens. When monocytes leave the circulation, they undergo a name change. So when they enter tissue, they are now referred to as macrophages or histiocytes. Lymphocytes are the other granulocyte. Lymphocytes, unlike monocytes, have very little cytoplasm. So when you look at a lymphocyte, basically what you see is a nucleus. You see a large, very darkly staining nucleus. And it's fairly uniform. Unlike the basophil, which was a big cell with lots of blue-purple spots, the lymphocyte is smaller, and it's just kind of like a uniform fuzzy purple. Not nice, distinct spots. Sometimes it's granular or grainy, but it's not, you know, like beach ball type spots. Also, lymphocytes have very little cytoplasm. Often you can't see the cytoplasm at all. But when you do, what you see is just this thin little rim of cytoplasm. Usually it's like a little crescent off to one side. Now, lymphocytes come in two different types. They can be small or large. So small ones are barely larger than a red cell, but the larger ones could be, you know, 10, 12 micrometers in diameter. But they still look the same. Now, lymphocytes come in two different types. We've got B cells and T cells. Remember, B cells are the cells that produce antibodies. When they do that, they become plasma cells, produce these antibodies. T cells are different. They don't produce antibodies, but they have various functions, and so they have various subclasses, like T killer cells, T helper cells, etc. We talked about them in detail. Unfortunately, in the blood, B and T cells look the same, so you can't use size as a distinction. Lastly, we have cell fragments in the blood, and these cell fragments are the platelets or thrombocytes. They are derived from the large cells in the bone marrow, and these large cells are megakaryocytes. So out of the peripheral blood, what we see are these little basophilic bits that look like bits of debris. And their job is to act as physical plugs in the event that there's some damage to the endothelium. Like you cut yourself and there's some damage to the wall of the blood vessel. These platelets will adhere basically like tire patches to repair the leak. If you were to look at a section of bone marrow, megakaryocytes are distinguished by the fact that they have a single multi-lobe nucleus, although the cells themselves are polyploid, and they are very, very large. Normally, the blast cells, the immature cells in bone marrow, are large to begin with. They can be up to 25 micrometers diameter, like twice the size of most cells in the body. But these megakaryocytes are up to like 100 micrometers diameter, so they're really big. So if you see a really big cell with lots of cytoplasm and this big, lumpy, bumpy nucleus, that's going to be a megakaryocyte. And the way these platelets form is by what's called demarcation channels. You get these basically lots of cytoplasm produced by the cell, and then extra bits of cell membrane. And the cell membrane starts to form essentially like perforations in the cytoplasm. And so these little bits of cytoplasm surrounded by cell membrane just begin to tear off of the edges of the cell. And these then are released into the peripheral blood as platelets. And that's just a picture of bone marrow showing these various stages of white blood cell formation. So that brings us to next to our major tissues, connective tissue. Yes?
We need to know those different stages, the white blood cells.
The question was, do you need to know those different stages of white blood cells? If you did, you are miles beyond anyone else in class. The answer is no. Even histologists or pathologists hate doing bone marrow biopsies because it's so difficult to distinguish with different cell types. Because all black cells look alike. They're all big and darker and funny looking. Okay, so connective tissue. What do we know about connective tissue? Well, the cells in connective tissue first are called fibroblasts. They are spindle-shaped cells. They're long and skinny. They've got a distinct nucleus, and they've got these little pointy ends. Now, the thing about them is that they produce extracellular products, this matrix, this amorphous matrix material, and they also produce various proteins as glycosaminoglycans, mucopolysaccharides, all that extracellular goo that forms the matrix. But they also produce various fibers within this extracellular environment. The most common of these are different types of collagen, but they also produce things like reticulin, elastin, and all that good stuff. So when you look at connective tissue, you have these cells that are not really loosely packed, interspersed with all this other stuff, which may be variously packed. And because everything stains pink, it's often difficult to distinguish the cell boundaries from the matrix itself. So usually what you see are these long spindle-shaped nuclei, which represent the fibroblasts. And then this matrix material is variously granular. And then depending on the packing and the properties of the extracellular matrix, you can often see darker pink fibers within the matrix. The most abundant of these fibers is type 1 collagen. It often packs fairly densely. And so here you can see these fibroblast nuclei, and here you can see these large collagen bundles, often forming these linear patterns. We see that in ligaments, bone, tendons. Type 2 collagen is restricted to hyaline cartilage and elastic cartilage. And so we'll talk more about those when we talk about the skeletal system. So type 2 collagen has a limited distribution.
Where is the elastic cartilage?
Where is the elastic cartilage? Well, for instance, over here, these dark pink fibers represent the elastic fibers. And then in hyaline cartilage, they're common around the periphery of the cartilage mass. But more about that later. Type 3 collagen is a specific type of collagen. It's also called reticulin. And unlike the other fibers, which tend to be long filamentous molecules, reticulin tends to branch. It forms like a net-like structure. And so we see reticulin in places like lymph nodes and bone marrow. And reticulin is also somewhat different in that it is R-gyrophilic. That is, it will stain positively with a silver stain. So when you form one of these silver stains, the reticulin fibers show up as a black, lacy network of fibers. Type 4 collagen is restricted to the basement membrane. It also does not form fibers, but instead tends to form sheets. So it forms like a felted meshwork that is used to support epithelia. So when you look at the basement membrane, that is composed of type 4 collagen. Elastic fibers are similar to collagen, although they have two basic structural components. They have elastin and fibrillin within them. Elastin makes up the core of the molecule, but it's kind of an irregular, sloppy arrangement, and we have on the outside these grooves or channels which are then filled in with fibrillin. And here you can see elastic fibers on H&E, these bright pink strands, and then here you can see them stained with a special stain on the right. So they're a little darker staining and more brilliant than collagen. Yes?
How are we supposed to differentiate elastic fibers and reticulin?
Well, elastic fibers, for instance, would be blue in a special stain. Reticulin fibers would be black on a silver stain. Reticulin fibers are thinner and branching. Reticulin fibers are thinner and branching. Elastic fibers are thicker and linear. So we have different types of connective tissue depending on the matrix qualities. So it can be loose and fairly open, almost like adipose tissue. It can be very dense, tightly packed, It can be dense and regular, where the bundles line up linearly, or it can be irregular. So different types of collagen. Adipose tissue, we've got white fat and brown fat. White fat is run-of-the-mill fat, which is said to be unilocular because it has a single large open space with very limited cytoplasm. Brown fat, on the other hand, is used for thermoregulation and things like hibernating animals. And it is multilocular, so it has a very grainy appearance to it, and comparatively more cytoplasm associated with it. So we get to epithelia. Remember, epithelia is supported by a basal membrane of type 4 collagen. On EM, it's got various layers that you can see. There's a lamina lucida, then there's a lamina densa, and then a lamina reticularis, which fades into the surrounding environment. With cell membranes that hold epithelial cells together have various modifications. These are in the form of cell junctions. You should be able to recognize a few of these, specifically desmosomes or maculoadherens. Basically, they're two dark black fuzzy lines side by side. These are on the lateral aspect of cells, so holding them together side by side. You can sometimes have what's called a hemidesmosome, or half of the desmosome, which is at the base of the cell, holding that cell or anchoring it to the underlying connective tissue. And so if you see something that looks kind of like a desmosome but is oriented horizontally, that would be a hemidesmosome. Yeah?
With the desmosome, how would we differentiate between going back all the way to the nuclear core?
Sorry?
Differentiate it going back to like the nuclear core. When it feels dark like that. Yeah, electron.
Nuclear envelope. That's the same.
Oh, to just a nuclear envelope.
Yeah.
Well, because it's such a really little bit, it's very dark, and the rest of it's light, so it's just a little tiny bit. But yeah, depending on if you didn't know the magnification, it would be almost impossible.
Okay.
You'd have to have some knowledge of what the entire span looked like. And nuclear pores tend to have a fuzzier open space in the middle, and then they'll be darker at the edges. But yeah, that could be really, the bottom line is I won't be showing you a nuclear pore yet. So if you see something that looks like this, call it a desmosome. If you see multiple clusters of cell junctions together, that would be a junctional complex where you've got like a couple of fuzzy areas all together. You can also have modifications of the cell surface. These would be things like microvilli, which are cytoplasmic extensions or projections of the cell surface. They contain actin filaments, which anchor into the cell cortex or terminal web. They can be distinguished from cilia because cilia are somewhat larger and they contain microtubules. When you look at microtubules on EM, they are hollow. So they've got a hollow center. They're sort of straw-like structures. And in cilia, they are arranged in what's called a 9+2 arrangement. They arise from a basal body at the very base of the cilia. So when you look at the EM of cilia, you can see these double lines compared to the single lines. And in the low-power view, cilia would look like that. And then there's different types of epithelia. I'm running out of time, so I'm not going to go through the types. You should be able to identify them or classify them if I ask you to. Same for ducts. You should be able to identify ducts. You should be able to recognize that salivary glands have different components, a serous and a mucus component. And you should be able to recognize that the serous component is more darkly staining, and the mucus acini, on the other hand, are almost clear. Sometimes you'll see these what are called serous demi-lumes, these little crescentic shapes off the sides of the mucus acini, and you can occasionally see duct scattered throughout the gland. Around the edge of the acini, you can commonly see these myoepithelial cells. They require a special immunohistochemical stain to see the detail. Remember, their job is to contract to help express product to the lumen. You can also see some very specialized types of glandular cells. These would be goblet cells, which are unicellular glands within an epithelium, which secrete mucus. They're generally large and oval shaped, usually clear. If you do a special stain for mucin, these light up as being bright blue. Very pretty. Muscle, we've got a couple of different types of muscle: skeletal muscle, smooth muscle, cardiac muscle. Skeletal muscle fibers, remember, are multinucleate.